Hi, I am Wahid. I am student of MS Biochemistry in Quaid-e-Azam University, Pakistan. The topic of my presentation is pharmacological evaluation of zinc dithiocarbamate derivative by bioassays. The contents of my presentation are given here. In introduction, I will start from synthetic organic compounds. Synthetic organic compounds are compounds which have carbon as their primary constituent. These compounds are synthesized artificially in laboratory to enhance their chemical or medicinal activities. Organometallic compounds are also synthetic organic compounds, but these compounds have a bond between carbon atom and metal atom. Organometallic compounds are of great importance because of their metal atom. And because most of the biochemical reactions occur in presence of metal atoms, such as certain enzymes require metal atom as cofactor to perform its activity properly. Zinc dithiocarbamate is an organometallic compound which has zinc as central metal atom, which is coordinated by two groups by the sulfur atom. The cytotoxic, antioxidant, antimicrobial, and antiviral activities of this zinc dithiocarbamate compound have already been reported. But now we have a novel derivative of zinc dithiocarbamate whose pharmacological properties are going to be evaluated, and I will share with you. The aims and objectives of my study is to evaluate cytotoxicity, to optimize its dose for in vivo study, and to evaluate antioxidant activity of this novel derivative of zinc dithiocarbamate, to evaluate its antibacterial activity, anticoagulant, analgesic, and antidepressant activities. To evaluate pharmacological properties of this novel derivative of zinc dithiocarbamate, certain biological assays were performed, which included in vitro assays and in vivo assays. In vitro assays included antimicrobial assays, cytotoxicity assay, antioxidant assay. To evaluate antioxidant activity of the compound, two assays were performed, which included total releasing power assay and total antioxidant capacity assay. In vivo assays included anticoagulant assay, analgesic, and antidepressant assays. The results of cytotoxicity assays are given here. The percentage mortality of different concentration of the test compound, which is novel derivative of the zinc dithiocarbamate, was evaluated. Saline was used as a normal control. The result shows that this derivative has significant cytotoxic activity as compared to the control. The results of antioxidant assays are given here. Two assays were performed, total antioxidant capacity assay and total reducing power assay. In both assays, ascorbic acid was taken as positive control, where DMSO was taken as negative control. Results showed that this novel derivative of the zinc dithiocarbamate shows significant antioxidant activity as compared to the control. To evaluate antibacterial activity of this novel derivative of zinc dithiocarbamate, antibacterial assay by dish diffusion method was performed. Three bacterial strains were used, which were Escherichia coli, Klebsiella pneumonia, and Streptococcus aureus. This compound didn't show any antibacterial activity against all of these three strains. For in vivo anticoagulant activity of this compound, anticoagulant assay was performed. Aspirin was taken as positive control. The compound shown the significant anticoagulant activity in comparison to the control. and its anticoagulant activity increases with the passage of time. To evaluate analgesic activity of the compound, analgesic assay was performed, and diclofenic acid was taken as positive control for this assay. The results show that the compound has significant analgesic activity as compared to diclofenic acid, and its analgesic activity increases with the passage of time. For in vivo antidepressant activity of this compound, antidepressant assay was performed, and in this assay, fluoxetine was used as positive control. The compound shown significant antidepressant activity as compared to the control, and its antidepressant activity increases with the passage of time. In conclusion, this compound has cytotoxic activity. It also possesses antioxidant activity, but it doesn't show any antibacterial activity against 3 bacterial strains which were used. It has also shown anticoagulant activity and analgesic and antidepressant activities. In future perspective, this compound can be used in future to cure cancer as it possesses antioxidant activity. It may be used to develop the combination therapy for effective cure of the cancer. It may be used for the development of the antioxidant drug without any antibacterial activity. It may also be used for the development of effective anticoagulant. It may lead to an approach to develop a new analgesic drug and development of antidepressant drug. These are references. In acknowledgement to my ever sportive parents, siblings, colleagues, whose enormous support has made me to achieve my objectives. Thank you.